Welcome back. This is lecture 8 and E443, 543, virtual tissue modeling methods. As always, the classes are live streamed on YouTube and available for viewing afterwards. And I appreciate your patience with me today, giving an online-only class. Today, we were going to talk a little bit about diffusion and decay with the goal of building up to a simple avascular tumor spheroid model. And we're going to have to do quite a few steps to get to that, and I'll walk you through all those steps today. We're going to have to learn a variety of both scientific and technical methods. So before we get involved in that, maybe I'd like to just quickly get a very, very brief 1 or 2 minute update from people about project status. Pedro, how are things going?
So me and Emma, we have an outline of what we want to do, and then we discussed together and we decided to build a simulation in pure Python. And we are meeting regularly every week to figure out how to do it, and I advanced significantly, and I can show you by the end of the class what I got. It's not the project itself, but I could start it in pure Python, and things are running, things are going forward.
Great. William?
So for right now, I'm still looking into the paper, so I might need a meetup with you and like talk more about the paper and stuff, so I can get it started with it.
So why don't you try to finish reading the paper and making notes on it, and let's meet by the end of the week.
Okay, yeah, that would be great.
Santu?
I'm actually studying about glioblastoma cells. So if after studying like few more papers, I'll meet up with you this week.
Okay.
Thank you.
So a few things about last time. We talked a little bit about how to adjust settings in Player when we're visualizing chemical fields. In particular, it's useful to know how to adjust the range. Fixed ranges are useful for some kinds of display. Auto-scaling ranges are useful for other kinds of display. The ISO lines can be quite helpful. The scale contours can be quite helpful too. I hope they're not crashing in 4.5 the way they were in 4.4. And another thing that we probably want to remember how to do is how to read values from a chemical field, get concentration field, and then the coordinates will give us the concentration at any point in the field. And this kind of approach where you scan across the field and plot the values can be quite useful. Sometimes it's nice to see the color in 2D or even in 3D. Sometimes it's nice to have a scientific plot that actually has the values at each point along a line. I want to come back to two fundamental concepts. When we describe diffusion fields, we often talk about the diffusion constant or the diffusion coefficient. But the things that we typically care about are time and length scales associated with the chemicals that are diffusing. In particular, the diffusion time is the time it takes a molecule to go a distance L. And that diffusion time goes like the distance squared divided by the diffusion constant. Depending on the dimension you're in, there may be a prefactor, a numerical prefactor, so it may be L squared over 2D, and so on. But nevertheless, the key thing is that the time it takes a molecule to get a distance L by diffusion goes up as the square of the distance. And that means that diffusion is a very efficient process over short length scales and very inefficient over long length scales. It's also true that the diffusion constants change a lot depending on the molecule. A small ion like potassium has a diffusion constant of about 2,000 microns squared per second. A typical small protein like green fluorescent protein has a diffusion constant of about 7 microns squared per second. That means it takes about 1 second to go a cell diameter. And if you think about distances in terms of 10 microns, which is about a cell diameter, that's a very useful concept. How long does it take for the chemical to get 1 cell diameter, 2 cell diameters, 10 cell diameters? So proteins diffuse in a period of about a second over a cell diameter. Large molecules like DNA, of course, have small diffusion constants, so they don't move very fast by diffusion. Typically, they have to be moved actively. Oxygen, like sodium or potassium, has a very high diffusion constant, but even for a fast diffusing species like oxygen, diffusion times become unreasonably long. 100 microns is probably a little bit too small a cutoff, but certainly a millimeter is about the limit over which oxygen diffusion really is effective. And that sets the scale of the body's capillary network. We built a linear gradient simulation and explored that a little bit. And one of the things we could do was to see how long it took the chemical to go across the field of view. And that's a case where plotting the value versus position is quite useful. If we look at a time of about 120 Monte Carlo steps, in this case, the diffusion constant was set to about 1. After 120 Monte Carlo steps, the chemical has diffused about 10 lattice about the square root of that. At around 2,500 Monte Carlo steps, the chemicals moved about 50 lattice the square root of that. And so we could actually see the slowing down of diffusion as we go to longer distances doing this simple linear gradient plot. If we want the chemical to get all the way across 100 lattice we need to wait at least 10,000 Monte Carlo steps, and even after 10,000 Monte Carlo steps, it's really not equilibrated. It takes about 25,000 or more Monte Carlo steps to get a relatively flat gradient if the chemical always goes from the left. Another thing that we played with a little bit, we didn't really have time to do all the exercises that were in the class notes, was looking at diffusion in the presence of obstacles. In reality, the environment inside of cells is very crowded, and small molecules don't diffuse freely. They have to get around bigger things. That slows them down to some extent. In addition, the membrane acts as a barrier, a selective barrier. It keeps water out partially, keeps glucose out partially. It keeps various ionic species in and out. And cells have a variety of ways of moving material into them and out of them. Some of them are passive, like ion channels. Some of them are active pumps that move desirable species either in or out of the cell using energy. And we played around a little bit with some very simplified simulations of impermeable membrane, a impermeable membrane with an ion channel. and you could see what the shapes of the chemical fields are under these different circumstances. And as you change the simulation, an interesting one to do, which we didn't do, is to have this ion channel be blocked sometimes and then open sometimes, and see how long it takes for the chemicals to start flowing through the ion channel. Today, we're going to be focusing on building up towards an avascular tumor spheroid simulation. We're going to be imagining a situation which is pretty common both in vivo and in vitro, where you start out with some cells. These cells are not going to respect the normal signals that tell the cell to stop dividing and proliferating. And so they're going to grow more than they would normally do. When they do that, they're going to form a cluster of cells, which is what a benign tumor is. Now, to grow, cells need, in particular, oxygen. They need glucose, both as an energy source and as a source of building the biochemical components cells are made out of. And they're also going to need growth factors, which regulate the growth of the cell. Typically, we are going to assume that there's a single limiting resource. In reality, there almost always are two, oxygen and glucose, a minimum. And so that we're going to assume that the rate at which cells grow and divide is determined by the availability of a single resource. And so we're going to have to be able to understand what determines the concentration of that resource as a function of position. And that's going to be determined by the diffusion of the resource outside of the tumor and by the uptake of the cells inside the tumor. We're going to have some simple model of how the cells grow and divide in response to that resource. And then we also have to make a few other notes. To survive, cells need a certain amount of oxygen and a certain amount of glucose just to keep themselves going. To grow, they need more than that minimum. That minimum that they need to survive is called the basal level of metabolism. And if they don't have more than that, if they have less than that, they're going to eventually die. The cells will die. If the cells have more than that, they potentially, not always, but potentially can grow and divide. When metabolic resources are limited, but not extremely limited, the cells will stop dividing and enter cell cycle arrest. It's called quiescence. If the metabolic resources fall too low, then the cell's basal metabolism will stop and the cell will die by necrosis. And in the case of real necrosis, the cells need to continually pump water out of the cell. Because cells have a high osmotic concentration, water is always leaking into the membrane, through the membrane of the cell. And the cell actually actively has to pump water out continually. It uses a big fraction of its energy budget to pump water out all the time. It's like a leaky ship. And if the cell runs out of energy, of ATP, then the water pumps stop and the cell swells up like a balloon. And if it swells up too much, it'll burst. And that's classic necrosis. If I look at a tumor where the limiting chemical is coming from the outside and is being taken up by the cells, I will see that the concentration of the limiting resource will be high on the surface of the tumor, will decrease as I go into the tumor. And so if I have a threshold, which says if my limiting resource is above a threshold, the cells proliferate. If it's some intermediate level, the cells stop proliferating, become quiescent, but don't die. And if it gets too low, the cells die of necrosis. Then I get a classic three-layered tumor in which at the very center of the tumor I have necrosis, then I have a relatively thick layer of quiescent cells, and then I have a fairly thin layer of proliferating cells on the surface. And we want to get to the point of understanding why that shape happens and how to control it inside of CompuCell today. I've shown these as spheres, but in fact, diffusion limitation, diffusion-limited growth can lead to quite complex shapes, in particular the fingered shapes that are associated with metastasis and spread of cancers. Here are two simulations. In the first one, we have nutrient-limited growth. This simulation, it doesn't stop when it runs out of space, so the simulation doesn't really mean much once it's bumped into the walls of the simulation. In the second one, the necrotic cells are actually shrinking and disappearing, and you'll see you get these very complex spatial patterns. Initially, you get concentric circles of quiescent and necrotic cells in the middle, proliferating cells on the surface. The necrotic cells disappear. That causes a mechanical collapse of the tumor, and that mechanical collapse leads to these complicated fingered patterns that are typical of diffusion-limited growth. We're probably not going to get all the way to this simulation today, but I'd like to get as close as possible to that, and I want to walk you through all the steps you're going to need to be able to build that simulation. One of the things we have to talk about is decay and diffusion length. We've so far modeled diffusion by itself. Very often molecules decay. Oxygen doesn't decay. Oxygen is, once you have oxygen, it's there, although it can certainly react with other chemical species. But most proteins, for example, have a finite lifetime, and they will decay over time. Depending on the particular molecular species, the lifetime might be anything from minutes to months or even years. There's some molecular species in your body that are created during development, which last your whole lifetime. So there are individual chemicals, proteins and other chemical species that last essentially your whole lifetime. But the typical thing that we're going to have to think about is the diffusion equation with the decay term. And one thing that we have to remember with decay is that unless something unusual is happening, we always have what is called first-order decay. The rate of loss of a chemical is proportional to the concentration of that chemical. And the reason for that, we can make a variety of assertions about why it is the case, but a simple one is to think about it this way. We can't have a negative amount of the chemical, and so the rate of loss has to be zero when the concentration is zero. And so that means the simplest way to make sure that that occurs is to have the rate of loss be proportional to the concentration of the chemical. Another way to think about it is to assume that the loss of each molecule is independent of the loss of any other molecule. And if you make that assumption, you also get first-order linear rate of loss. If I model diffusion with decay, and I start out with a spike of chemical at a given position, I will get a classic Gaussian spread of the chemical concentration. The maximum concentration of the chemical will decrease in time exponentially. The width of the chemical distribution will increase in time. And the key thing is that if the decay constant is gamma, the lifetime of the chemical is 1 over gamma. And so we have to remember that the units of the decay constant are per time. So 1 over the decay constant is the lifetime. Once we have a concept of a decay constant, we can ask the question, how far does a chemical get before it decays due to diffusion? and that defines the diffusion length. The diffusion length is the distance over which the concentration of the chemical decreases by a factor of 1 over E, that is about a factor of 3, and that diffusion length is given by the square root of the diffusion constant divided by the decay constant. So the diffusion time was length squared divided by D. The diffusion length is D square root of D over gamma. Typical diffusion lengths for proteins in tissues are somewhere between 5 microns and 150 microns. That means that chemical signaling by diffusion typically cannot build structures that are bigger than a millimeter across. And if you think about our bodies are a lot bigger than a millimeter. And so that tells you that diffusion by itself cannot be defining the large-scale structures in the body. Again, we should think a little bit about oxygen and glucose from capillaries in the body. Concentrations of oxygen and glucose are high at the surface of blood vessels, at the surface of capillaries. They're both used up by the tissues that surround them, so their concentration decreases as we go further away. And that rate of decrease, again, is going to determine the separation between capillaries. And we're going to find that the typical diffusion lengths for oxygen and glucose in tissues are a few hundred microns, which means that you have to have a capillary about every millimeter, every half a millimeter in the body. And so our bodies have to have a very dense web of capillaries to function. In embryos, these diffusion lengths define the shape and size of primordia that build organs. You almost always start building organs with structures that have the length of typical length of a diffusion length or two. They're going to grow through all sorts of different processes, but the initial sizes are going to typically be of that size. In adults, gradients of these diffusible chemicals instruct stem cells when and how to proliferate. And that means in general, not always, there's some exceptions to this, but in general, if you're going to have tissues renew themselves, you need to have stem cells within a few diffusion lengths of the tissue damage region. Now, cells can move actively, they can migrate actively, and in that case, they can go distances that are quite a bit further than their diffusion length. And absolutely, in tumors, the presence of these gradients drives the evolution of cancer invasiveness and resistance to chemotherapies. One of the problems in an avascular tumor spheroid is that if I put a chemical to kill the cells, it doesn't get that deep into the tumor, so the cells in the middle don't die. And so that can be a problem there. The last of the three properties of diffusion that we have to worry about is the simplest one in some ways, and that's called the equilibration time. The equilibration time is actually just the decay time again. It's the time it takes the field to forget its initial configuration, and that's 1 over gamma. If I have no decay, then the initial configuration is never forgotten. One result of this is that we almost never actually run computations without some decay, even a small decay, because if we do, we're always sensitive to our initial configurations. Okay, so we have the diffusion time, which we saw earlier, distance squared divided by d. We have the diffusion length, which is the square root of the diffusion constant divided by the decay constant. And we have the equilibration time, which is 1 over the decay constant. Those are the three fundamental properties of any chemical field. Not the diffusion constant and the decay constant by themselves, but the diffusion length, the diffusion time, and the decay time, or the equilibration time. So let's start out by doing a simple simulation. We've done diffusion without decay before. Now I would like you to build a simulation, use the simulation that we had from last time, or 2 times ago. And this time I want you to turn the global diffusion constant, the global decay constant on. So I want people to start CompuCell. I'd like you to boot up the simulation you did for the linear gradient. And now, instead of having a global decay constant of 0, use a global decay constant of 0.001, 1,000th. Okay. And I'd like you to plot the concentration as a function of position using the code you wrote 2 weeks ago. And then I would like people to compare what happens when the global decay constant is 0 to what happens when the global decay constant is 0.001. If you wanted to get fancy, you could have two chemical fields, one of which had the decay constant, one of which didn't, and plot with the same diffusion constant, and then plot them both together. Or you can simply run two simulations and screenshot the concentration as a function of position and compare it. Once you have a global decay constant, then you can actually check whether the diffusion length really is square root of d over gamma. And you could check if the diffusion time really is what we said it was, L squared over D. So why don't people take a few minutes and try that out?
I have a problem from the code with last time because it just crashes. I'm not sure why.
Okay, do you want to screen share it?
Sure. I think it's the right code. I'm not sure. But anytime I try to run it, it just crashes. I tried turning things off. It's not fixing.
Usually when code crashes outright, it means there's a typo in the XML. Does it crash player disappears when you try to run it?
That's all show.
Usually if player disappears, it's almost always a typo in the XML, like an extra closed bracket or a missing closed bracket. That's the problem with the parse. Let's see. Okay. So when you try to run this, it just disappears on you. Is that right?
Yes.
Okay, let's scroll up and let's see if there's somehow we clobbered the diffusion solver. Okay, Potts looks all right. Diffusion solver FE, diffusion field 1, okay. Diffusion data and diffusion data looks okay. Well, you almost certainly don't want that initial concentration, although that shouldn't be causing you the problem. Okay, secretion data looks okay. Boundary conditions look okay. That looks all right. So you want to get rid of uniform initializer since you're not using it. And let's see. Constant value looks okay. Can we scroll up and just let's see if there's something else that's... Can we get rid of that initial value for in line 47? I'd get rid of... Okay, make it 0 perhaps. Okay. And let's try saving that and seeing what happens when you run it. Okay, well, it looks all right.
Thanks.
What you don't have is the plot of the concentration versus position. Do you remember how to put that into your Python?
I have it somewhere else. I can look for it.
Well, let's just walk through it together. So in the start function, you'd want to create a plot, create a scientific plot. There you go. And you just need one, could be concentration versus position. Probably the Y will be concentration and the X would be position. Okay. And you only need one plot series. Okay. And you might rename it concentration or something like that. Okay, and now in the step function, you're not going to iterate over cells because you don't have any. But you want to first get a reference to the plot, to the concentration field. So go to the CC3D Python, chemical field manipulation, get field value. Okay. Okay, you'll need to get field reference first. Sorry about that. So the same, go back to CC3D Python, chemical field manipulation, get field reference. Okay, good. And now that would be whatever the chemical field name you used was.
Maybe the all caps are going to be.
No, in this case, it's not. It's only, the all caps is only for the names of cell types.
Okay.
And I really don't know why we did that. It seems bizarre to me that we made that choice. Okay. And now what you want to do is you want to iterate from left to right through your simulation. So do a for loop for X in range 0 and then the size of the lattice. Is it a 40 by 4? Is it a 0? Is it a 40 0 by 40 lattice? Yeah. Okay. Fine. Great. Okay. And then you want to plot the. So you want to add a plot data point. Good. And so be concentration. And then the X position would be X. So instead of MCS, it would be X. And then Y, you need to read field, square bracket, 10, sorry, X, comma, 10, comma, 0. Okay, that should do it. See if that works. And get rid of line 40. Okay, so here we're just going to see if that'll run. Okay, now let's see if we run it, what happens. Now what's happening is that it's drawing a lot of lines that we don't want, so we probably want points rather than lines in our plot. So we want points there. And we probably don't want to plot that every Monte Carlo step. It's too frequent. So maybe every 10 Monte Carlo steps. So okay, great. You're missing a column. Okay, great. There we go. Were other people able to get that to work now? Does anybody need help with that? Yeah, self.dim.x will tell you the size of the simulation so you don't have to remember it. That really should be in the Twit code snippets. That one I always have trouble remembering. Okay, thank you. Okay, were other people able to get that to work? Does anybody else need help with that? Okay. So if we compare the case with global diffusion constant and without, if I wait a long time without a global diffusion constant, with the global decay constant without, if I run for a long time without global decay, I get a linear gradient. If I run it for a long time with the global decay, I'll get an exponential. And maybe we didn't really wait long enough to see that we reached not a linear gradient, but an exponential gradient. So that's something that is worth looking at. The spatial length scale of that linear gradient, of that exponential gradient, will be given by the square root of the diffusion constant divided by the decay constant, diffusion length. Okay, so that's the basic idea of decay. And now we're going to generalize the idea of decay to sources and sinks of chemicals. And so in diffusion equation with decays and sources, we're adding a term S, which is a source of the chemical. Clearly, the chemical has to come from somewhere. So far in our simulations, the chemicals come from the edges of our domain. And that may be realistic if we're simulating something like oxygen coming from the top surface of a culture dish. But in general, in biology, oxygen comes from a blood vessel, for example, or from the surface of the lungs, not from some distant external source. Something that is worth remembering, and I think this is actually Pedro's slide originally, is that if there's a source and decay happening at the same time, and the source is a constant rate, which is shown here, then the equilibrium concentration is given by the source rate divided by the decay rate. And actually, this should be gamma, not C. There are a variety of biological ways that you can have sources and sinks. And they have somewhat different behaviors depending on which ones are going on biologically. Secretion can happen through membranes. It can happen through ion channels. It can happen through pumps. It can also happen through exocytosis, where you actually package chemicals in vesicles, and those chemicals then are released when the vesicles fuse with the cell membrane. These have somewhat different properties. If you have diffusion across a membrane, then the speed at which you diffuse across the membrane, the rate of the source, is dependent on the difference in concentration just inside and just outside the membrane, according to what's called Fick's law. If you have secretion by vesicles, the rate at which vesicles are produced and pumped into the outside world is essentially independent of the concentration outside the cell. And therefore, you really have a constant flux. The rate at which you're producing chemical is independent of the chemical concentration outside. In the case of things like blood vessels, the oxygen concentration is essentially fixed to a constant value at the surface of the blood vessel. And so in that case, you have constant value boundary conditions. And so you can have constant flux boundary conditions, Fick's law boundary conditions at which the rate of flow is dependent on the gradient across the membrane, and constant value boundary conditions. All of these can be thought of as secretion of one sort or another. Both secretion and absorption in CompuCell can be implemented in a number of ways. They can be implemented in the diffusion steppable itself. There is a secretion steppable and perhaps it's a little bit misleading, that the secretion and absorption are both implemented through something called the secretion steppable. So if you're doing uptake or absorption, you use the secretion steppable. So you have to remember that the name isn't quite consistent. If we're going to implement secretion, we have to decide which cells or what kind of other objects besides cells are doing the secreting, where, with respect to the cell, the secretion happens, whether secretion happens everywhere or only when the cell is in contact with, for example, cells of a particular type or medium. In epithelia, very often cells secrete out of their top surface. The mucus that's accumulating in my lungs is being generated by the goblet cells in my lungs that secrete mucus out of their apical surface into the alveolar sacs. Similar things will happen in your gut lining, for example. And we need to know whether we are secreting a constant flux, that is a constant rate of secretion, which would be typical of exocytosis mechanisms, or a constant value at the boundaries. Again, secretion and absorption are handled computationally through the secretion plugin, and we're going to need to load that in the markup language, either through the diffusion solver or as a freestanding plugin. Typically, we're going to load the plugin by itself. We control it from Python. As usual, if we control it, if we declare it, the details in the XML, then we can't do the full control in Python. We can have multiple sets of secretion rules for the same cell or the same field. We have some examples of that in the demos for today. Let's look at what the code looks like. If we're going to define secretion in the diffusion solver itself, there is a block called secretion data, and I can say that we have secretion by cells, in this case, cells of type bacterium, secrete, and the number by itself means that you're secreting at a constant rate. I could have secretion on contact with, in this case, when bacteria are in contact with wall, I secrete at a constant rate. I can secrete at a constant value, constant concentration, at a given type. So those are possibilities. There's also a secretion plugin separately, a plugin named Secretion, and I could do the same things that I just did before with some more flexibility, and I'm going to go over all of the different permutations. I don't expect you to remember all of them, but it's important at least to know that they exist so that you could look them up. Typically, we're going to use the secretion plugin bare and then do all the specifications in Python. We have a couple of basic concepts: constant rate secretion, constant concentration secretion, and secretion on contact. Unusually for CompuCell, if you specify secretion in the diffusion solver and the secretion plugin, they both execute. They'll add on top of each other. The one in the secretion plugin will be executed temporally first in the sequence of execution in the code. And if you actually specify it in Python, it will not replace what was specified in the XML. It will add to it, and that will be executed third. So you can actually specify secretion and absorption three ways, and they add on each other. Okay. If we're going to do secretion through Python, we need to specify the secretion plugin, the XML. We have to tell the secretion plugin which field we're talking about. So we're always going to have a get field reference when we use secretor. Then in the step function, we need to use that reference to the field and tell CompuCell how much chemical to secrete. And this is on a per voxel basis. And that means there may be some scaling issues that we have to think about. And there are quite a few options. We could specify which cells we want to secrete, all cells of a given type, individual cells. We can have cells secrete inside the cell at the boundary, just inside the surface of the cell. Inside the cell at the boundary when it's in contact with another cell of a given type, for example, for epithelia. Inside the cell at the center of mass of the cell at a single voxel. Inside the cell everywhere, not just at the surface. Outside the cell, just outside the cell at the surface. And outside the cell at the boundary on contact with another cell type. So there are 1, 2, 3, 4, 5, 6 different loci where we can secrete. And it's quite convenient to ask the secretor, and it's even more convenient when we're doing absorption, to ask it how much we secreted. And so there's a total amount option that asks the secretor to calculate how much chemical was in fact moved per time step. If we are secreting inside the cell, we could specify constant value secretion rather than constant flux. You could say, why don't we have that for all of these? And the answer is, if you think about it, not all of them make sense for constant value. Okay. Syntactically, if we're going to use the Python specification, we need to have in the XML the bare plugin secretion plugin. Then in the step function, we are going to get a reference to the chemical field, self.get field secretor, and then the name of the chemical field. And we only have to do that once. Typically, we do it once per step in the step function, but in fact, you could do it once in the start function and pass the reference to the step function if you wanted to. The field secretor object is a complex object, and something that I recommend getting in the habit of doing in general with a complex package like CompuCell is to use the dir, the dir function, on objects. If you print dir of an object, it'll give you the object structure and all the methods and attributes associated with it. If the object is something very simple like a float, it won't be very exciting. But for complex objects, you don't always know what are the things I can do to it. The manuals help, but sometimes printing a dir is the fastest way to learn what you can do. In this case, when you print the secretor object, you'll see that it's quite a complex set of options. Okay. Again, if I want to know how much of a chemical there are a number of kinds of questions that I might ask. I might want to know what's the total amount of chemical in the chemical field at any given time. That is given by the method total field integral, which then I can apply to the secretor. And that will return to me the total amount of chemical in the field. If I want to know how much chemical is within the boundary of a given cell, which is also a pretty useful thing to be able to do, I have a method called amount seen by cell, which operates on cells and the chemical field. And so it's important to remember these two functions when you're designing simulations. Okay. As I mentioned, you have a lot of different options for doing secretion. When I'm going to do secretion, I take my secretor object, whatever the handle for my secretor object was. Here I called it ATTR_secretor. And then I apply to that a method here, secrete inside cell at boundary. Which cell and how much? Again, I have lots of options for how secretion is going to work, and I'll walk you through those quickly. Again, you don't have to remember all of them. They're all in the XML, in the Python code snippets and in the manuals. But it's useful to see what they look like once so that you can familiarize yourself with them. Total count is a useful thing. It will tell you the total amount secreted. And you have to be a little bit careful because the return from total count is not a number. It's in itself a complex object. And so the actual amount is res.total_amount. Again, if you don't remember this, get in the habit of printing the dir of the object, and that'll tell you all of its attributes and how to reference it. Any questions about that so far? So let's go through the possibilities. If I want to secrete just inside the cell membrane, and this would be typical of cells which have a transmembrane diffusion, for example, I'll get a reference to the secretor, ATTR_secretor. Again, that handle could be anything. It's just a reference. Get field secretor and the name of the chemical. Here I called it ATTR. Once I've gotten that reference, I could secrete inside cell at boundary, an amount of 300. I could secrete inside cell at boundary total count. That'll return the number 300 because that's what I told it to do. I could secrete inside cell at boundary on contact with wall or medium. Here, and for example, I want the cells to only secrete on their top surface. So I'd say secrete on contact with medium. And I can keep track of how much there is as well. Okay. If I'm secreting by exocytosis, then the secretion doesn't happen just inside the membrane, it happens just outside the membrane. And so the word inside is replaced by outside everywhere. Secrete outside cell at boundary, secrete outside cell at boundary total count, secrete outside cell at boundary on contact with. There are a lot of permutations, but they're pretty straightforward once you figure out what the structure of the commands is. Especially if I'm imagining my cell being a 2D surface of a three-dimensional layer of cells, so I'm looking down from the top, then I might expect that the cell to be secreting everywhere along its surface. In that case, I would be likely to use secrete inside cell as my secretor. And again, I have the option secrete inside cell total count, secrete inside cell. And here I have an option that I haven't seen before, constant concentration. Constant concentration. In this case, I'm not putting a given amount of chemical into the extracellular space. I am forcing the concentration in the field to be that value everywhere in that region. Again, that would be typical of what happens if I have a blood vessel, for example, which is holding the oxygen concentration in the tissue at the surface of the blood vessel to be a particular value. To agree with other simulations, because it's pretty common in other simulation methodologies, you can tell the system to secrete at the center of mass of the cell. And there are a variety of reasons that I don't particularly recommend doing that. It tends to give you rather noisy simulations, but it's pretty common. And so if you're trying to replicate a simulation that was done with that, it's important to have it available to you. Okay. So now, why don't you try this? for yourself. If you don't remember everything, go look in the manual, the Python scripting manual, or the CompuCell reference guide. Or for that matter, you could maybe try ChatGPT and see if it'll give you the code. I want you to create a simulation. You could use the simulation you just used, actually. But create a single frozen cell in the center of the simulation. A single chemical field, you could call it oxygen if you like. Add the secretion plugin, just the bare secretion plugin in the XML. Set the diffusion coefficient to 1, the decay constant to 0.01. Have periodic boundary conditions for the diffusion field. And then have the cell secrete with uniform secretion and constant concentration secretion. And use the code that you just wrote to plot the concentration of the chemical field as a function of position going through the cell. Okay? Is the exercise clear? Any questions about the exercise? Santu, I'm going to ask you to show us your results in a few minutes. So if you need help, please reach out maybe to Hayden or to make sure that you've got this working. If you got this working, I'll give people about 7 minutes to try this based on what we already have. There is a version of this code in the student materials folder, by the way, if you're not able to get it on your own, but try to get it on your own. And if you've already done the basic exercise and you want to try to do something a little more exciting, use different secretion options. Inside cell at boundary, inside cell at COM, inside cell constant concentration, outside cell at boundary, and compare the plots of the concentration for the different options. Okay? Okay. That looks great, Alex.
My only issues with the plot. I'm not sure how to do it, but
So let's look at the Python for a moment. See what we've got.
it was crashing. Or it wasn't crashing, it was just giving an error.
Well, so I can see one thing. You're not iterating over the position in this case.
Probably why. Oh, that's what I was forgetting. Yeah, that makes sense. I knew this. I think I didn't like X. That makes sense.
So there are two things that I will comment on. One of which is if you look in your code. Sorry about that. Let me get this set up. If you look at your code, when you get the reference to the field secretor, line 28, you want to move that outside of the for loop. You only need to get that reference once. Okay. And the next thing that you want to do is when you're plotting, you don't want to be plotting for the cell, so you want to move that outside of the for cell loop. But you do need, just the way you did for the plot you were doing before, you do need to scan across the position in X. Yeah, there you go. And in this case, the size is 100, so you need to make that. The code that if you the size of the simulation is available to you. And that's, as I say, this is one of the things that probably should be in Quidit that isn't, which is if you use that self.dim.x, then when you change the size of the simulation with an I, dim, and probably when you're doing field, you probably want to scan halfway across. So instead of x comma 10 in the next line, x comma 10, you might want self.dim.y divided by 2. That'll go across the middle. Why don't you give that a try? There you go. Now, tell me why the concentration is lower in the middle of the cell than at the edge. Anybody? What was the secretion rule that you used? Inside cell at boundary. Okay. Does the chemical decay? Do you have decay in the chemical?
I do not think so. I might.
Do.
Maybe I do.
Yeah, you do. 0.01.
I forgot.
So that means that the chemical concentration will be maximal at the surface and go down inside and outside. So what you're seeing makes sense. What if we try a different secretor? What if we try inside cell at COM instead? What do you think it'll look like this time before you do it? What shape do you expect? Where will the concentration be maximal?
Maybe at like in the very middle of the cell.
Perfect. Great. It's always a good idea to test yourself and try to figure out what you expect. So let's see what happens. Yep. And you'll notice how spiky it is. It's really very, very big right in the middle and then goes down rapidly. That's why I say that secreting at the center of mass isn't that great an idea. You get very spiky chemical concentrations. What about inside cell at constant concentration? Let's try that one. Yeah, if the people who have Mac problems are getting a message like cannot verify, please screenshot that and we'll send that on to our tech team. Okay, so what happened? What do you expect for this one? Here, the concentration is flat. It's the same everywhere. And you'll notice before the concentration kept going up and up as I was secreting more and more. Here, the maximum value of the concentration is fixed. So thank you very much. That was great. Appreciate that. Okay. So in my case, I created a single cell in the middle. I had, you could do it with secretion inside of the diffusion solver, but in this case, we were doing it with the XML. The example of
What the fuck?
constant value we just saw, constant concentration we've just seen. And we tried some of the other exercises that one could do as well.
William, your mic is open.
If we try to play here a little bit, we could see a variety. We could see a variety of different patterns. Inside cell will give us a maximum at the center and a fairly smooth profile. Inside cell at boundary will give us spikes at the boundary, minimum in the middle of the cell and outside. Center of mass gives us a single spike at the top. Constant value gives us a flat top. Inside cell and outside cell at boundary look almost the same, but the position of the maximum is just inside or just outside of the cell. Okay. Now I'm going to talk about uptake. And one would think initially that uptake would be exactly parallel to secretion. But just the way when we do decay, we can't have the concentration go negative. We can't take up something that isn't there. And in reality, the rate of uptake of chemicals by cells is a very complex function of their concentration in the environment. Cells may take up material by endocytosis, the opposite of exocytosis, in which case they take up essentially constant amounts per unit time from what's available. They can pump or have diffusion across the membrane, in which case they have constant transport rates. And in general, the amount that's taken up is really a bit of a hill function. If the concentration is low, you take up an amount that's proportional to the extracellular concentration. But very often for a lot of these processes, like endocytosis, there's a maximum uptake rate. And so there's a saturation. Something that you might not expect is that the field profile around the cell may not actually do what you expect it to do, at least might not be so obviously what you expect it to do. In particular, you'll find that if a cell is taking up chemical, the concentration field will start decreasing quite far from the surface of the cell. It won't decrease just at the surface. Again, we can simulate uptake in a variety of ways. You could have a decay. Decay effectively behaves like uptake. So you could say that I have a particular decay coefficient in a cell of a given type. If I use the uptake function, it has not the full complex uptake rules that I just showed. It has a simple uptake rule, which is you have uptake that's linearly proportional to concentration below a maximum, and then a maximum uptake rate. And so the secretor for uptake will have all of the same options that we have before. Inside cell at boundary, outside cell at boundary, inside cell at center of mass, total count, which in this case is more meaningful perhaps than on secretion. And the arguments are the cell ID, sorry, the cell handle, not the cell ID, the cell, the maximum level, and the fractional amount. And to be honest, I think the order of those is a little bit confusing. If I were going to do it again, I'd put the relative amount first and the maximum second. Another thing that you're going to notice is that when you get total count back on uptake, it'll be negative. And so if you want to know how much you actually absorb, you're going to have to put a minus sign in front. In a lot of situations, the cells take up molecules just inside the membrane. And so inside cell at boundary total count is a typical thing to use. In reality, the rate of uptake should be proportional to the concentration difference between the inside and the outside. And that's not available. We can do that by hand if we need it, but we do not have that as a primitive operation in CompuCell at the moment. In many cases, uptake occurs only on certain surfaces of cells. And so we have the option of uptake on contact with to simulate things like absorption of chemicals in the gut, for example. And so we have all of the same options that we had before. Inside cell at boundary, outside cell at boundary, inside cell at boundary on contact, outside cell and boundary on contact. Everywhere in the cell, which is the one we use pretty often. And then again at the center of mass, which is not something that we're going to use very often. It really doesn't work very effectively, although we're going to find that if we're trying to replicate published results, it's a pretty common one. So I'd like people now to take the simulation that they just had. In this case, you're going to have to start with some amount of chemical in the environment. So you can set the boundary condition on the chemical field now to be 10 in both X and Y. So you have chemical coming from the boundaries, constant value boundary condition. And then instead of doing a secretion by your cell, do uptake by your cell. And try inside cell at boundary, inside cell at center of mass, and outside cell at boundary for uptake. And see how those compare. And if you get that one done as a stretch exercise, instead of having the secretion come from the perimeter of the simulation, have medium secrete at a constant value of 10 and see what happens. Go to player. Okay. Okay. Type twit it. Fine. Launch them. Okay. I'm sorry, what are you doing? It's twitted is there. Yes. So you've got twitted there. Okay.
Yes, sir.
So now please launch the code that we have. Okay, do you want to start from scratch on this? That's okay. If you prefer. Why don't you tell us what you're going to try to do before you start doing it?
I'm just starting with the diffusion, that's it.
Okay,
With the diffusant and all.
okay, that's great. So why don't you go back a step and you could make the lattice 100 by 100. There we go. Great. That's fine. And you want your cell to be frozen. So hit clit freeze. There we go. And hit next. Good. And now you can create a chemical field. Perfect. Okay, you don't need any of those. Don't. But you do need, no, back. You go back. 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 You're going to need the secretion plugin. There you go. Okay. There you go. And you could just leave that empty for the moment. Okay. All right. Hit finish.
Yeah. This is what is happening with the Reddit.
I see. So it's crashing there. Which version of CompuCell are you running?
Um, the latest one, I guess. I just reinstalled it recently, so
Okay.
yeah.
So please. Okay. We don't have to do it this way. Open it again. Open Twitter again. Please go up to help and select help at the top. Intuit it and hit about. Okay, that is the recent one. Okay, fine. So I suspect that it's failing to create the file because you're probably pointing to a directory that you don't have write privileges for. Did you try downloading the code from the user directory that I sent before?
Yeah, yes, sir, I did.
Where is that code?
Just like,
Okay, good. So why don't you move? No, you don't want just, you can't just have CC3D. Where is the, it's a whole, we need the whole file, the whole zip file. You don't have that? Let
uh,
me get that for you again. You don't have it.
okay.
Okay, good. So now open that file that you just downloaded. No, not new. Hit open on all the way on the left. There you go. Open CC3D project. Point to the file you just downloaded. There you go. That one. Open. Hit open. That's fine. It's okay. Okay. Now you may have to hit open again. So try opening it again. Okay, now go to exercise 9_1 single secretor. Oh, don't.
Oh.
There you go. Open that. Open it again. And you put it inside of another. Okay, open simulation. Huh. That's strange. Pedro, is there a problem with that folder? I may
I
have
don't think so.
given you a defective folder. Let's see. No, it's okay. So it's not that. All right, let's do it by hand. Go back up to downloads. Sorry about this. Go back to downloads. Open that file there. Open it again. Huh. Okay, where is the zipped file? Where did the zip file go that you downloaded?
Just give me a second, sir. I'll download it again.
Download it again, please.
That was an extraction menu. It wasn't showing the .cc3d file because it was an extracting explorer. It doesn't show any file, only folders.
All right, so we can unzip it if we need to. Did you download it again? I don't think so. Use the link. No. Please wait and there you go. Fine. So unzip that. Unzip exercise 9_1 single secretor. Unzip that. No. go to the compressed file and unzip it. Right click on that and hit unzip. No, not using Twitter, just by hand.
Yes, yes.
Just unzip. Extract all. Okay. Don't use a give. Okay. Fine. So now move that folder onto your desktop. Okay. Open that folder. Where did you put it? There we go. Okay.
Yes.
Open that folder. Okay, fine. Now use Twit. Now use Twit. Okay, don't unzip. Just hit no. Okay, I'll hit open. Fine. Now point to now point to your desktop. No, to your desktop. Okay, now open that. Hit, it's there. Open that. Okay, now double click on exercise 9.1. Okay, fine. Now hit run. Open the simulation in player.
Yes, this is blocking it. Okay.
No, it's not. Just hit open simulation in player.
You right click under CC3D simulation on the left. Right click on exercise 9-1. And then open in player, second option there.
Okay. Was anybody able to get the uptake to work? Alex, did it work for you?
I think so.
Shall we take a look?
Yeah. I started doing with the medium secretion.
Now, with the medium secretion, it's sort of a more interesting problem. So let's see here.
Code first. Saw that one.
So one thing you're going to have to do with this is, let's look at the XML for a minute. Is you're going to have to use, okay, you have min value X and Y. You want to also have that at max. So copy 72 and make it max as well. And the same thing for 80, 81. Yeah, there you go. Try that one. Now you've got medium secreting as well, so you've got it duplicated, but let's comment the medium one out for the moment. Okay, try that one first. Let's see what you got. Yeah, so now you get a nice inverted U-shaped concentration. I noticed that the concentration goes down all the way from the periphery. It doesn't just go down near the cell. I think that's the key thing to notice here. Okay. What if we change the uptake to be inside the cell at the center of mass, for example? So that would be in the, there we go. It takes a while for the chemical to get to the center to be seen in the first place. Looks like you have two kinds of uptake going on, perhaps. Let's take a look. You have inside cell at center of mass. That looks fine. Let's look at your XML and see what you've got. I see what's going on. You have you have decay inside the cell turned on as well. And the diffusion constants are not the same everywhere. So you want to make diffusion constant of 1 there is fine, but global diffusion constant should also be 1. You don't want a global decay constant. And you don't want a decay coefficient inside the cell. Fine, there we go. I mean, the things that you have there are not wrong, but there are additional factors that are making it harder to understand what's happening to the simulation. So here, really, you're uptaking at a point, so you could see what it's doing. Then you could also see why that might not be something that's biologically very meaningful. Okay, why don't we go back and try inside cell then and see what we got. So now that makes perhaps a little bit more sense, I think, what you're saying. Now here you're taking up if you look at the amount of chemical versus position, you'll see you're taking up almost all of it, right? If you look at the amount in the middle, it's very small. So what if we made the maximum uptake amount less? We could make the maximum uptake amount something like 0.1 instead of 0.2. You have to change the first one is the max, so you have to make that one smaller too. I might even, well, okay, making it 1 is all right. I'd make it 0.1. Try making it 0.1 as well. Okay, there. Now try that. We're still taking up most of the chemical in the center, but now it's a bit smoother. So this is something that really is worth exploring, and I'll try to write a little homework assignment for it. Because understanding all, thank you very much. That was very helpful. Understanding all of the different configurations that are possible and how they interact with each other for uptake is actually not trivial to do. So it does take some practice. Thank you, Alex, for being the guinea pig for so much of class today. Okay. So we did, this is almost exactly what we did here. I put in a global decay constant, a weak global decay constant. And you get the kind of configuration that we just saw. Okay. When you play with this, you can get a variety of different shapes, depending on whether you uptake inside cell, outside cell, center of mass, at boundary, and so on. Something that could be pretty interesting, and Alex, I think you were just starting on this, is if you have secretion in the medium so that the concentration in the medium is fixed. In that case, you'll see a constant concentration of the chemical in the medium, and then a change of concentration only within the cell. And you'll get, again, quite different profiles depending on whether you have secretion uptake inside the cell at the boundary, inside the cell at the COM, and so on. So I'd like to come back in our last 45, 50 minutes. I'd like to come back to the avascular tuber spheroid. We now have the ability to secrete chemical. We have the ability to take up the chemical. And now we have the ability to measure how much of the chemical we took up and change whether the cell is going to be proliferating, quiescent, or necrotic based on the amount of the chemical that we took up. And what we're going to start out by doing is having a very simple rule that says that if we imagine, for example, that the chemical is oxygen or glucose, that if the cells have a lot of oxygen or glucose, they proliferate. If they have an intermediate level of oxygen or glucose, they become quiescent. And if they don't have enough at all, they die. They become necrotic. And something that's a biological observation is that if cells become quiescent and you put them in more favorable environments, they can go back to proliferating. So proliferation to quiescence and back is reversible. Once you get to necrosis, the cell is definitively dead. And so you can't go from necrosis back to quiescence with proliferation. I had a few comments which may be worth saying a few words about. Something that we saw when Alex was coding was that we were changing values. For example, we were putting the size of the lattice into the simulation. And in the case of the lattice, we can reference the size of the simulation, self.dim.x, self.dim.y. But in other cases, for example, the time step or the length step, those aren't built-in functions. And it's useful if we're going to have a lot of parameters, and we might have a lot of parameters in a cancer model, for example, to try to put them into someplace systematic, not just put them in the code where they occur, but have a block of code which defines the parameters. And it's also useful if you can, and there are some reasons it's not so easy with the XML, to try to have some scaling factors. For example, a statement that one Monte Carlo step is 1 minute or 60 seconds. A statement that one lattice site is a micron, for example. And then define the other parameters with reference to that, so that if you change the length scale or the time scale, the other parameters change automatically. As a note, when you're annotating parameters, you should always add a comment that specifies the units of the parameter. You have a number of places you can define these things. You can put the parameters into your Python code before all of your class definitions, at the very beginning where you do your imports. And that is convenient because the parameters are available everywhere. They really are parameters. You cannot change their values. If you try to change the values, you'll get an error. But I could say from CC3D, import blah, uptake rate equals 0.1, noting that that's fractions per Monte Carlo step, and then uptake rate will be available in all of my classes. If you want parameters that can be modified, you can use the global dictionary self.shared steppable variables and create a dictionary entry and put it there. So that's another possibility. And that's available anywhere. And those can be modified if you need to. If the parameter appropriately references a steppable, then you could put the parameter definition with a self dot inside the steppable declaration after the init. Self dot quiescence threshold equals 7 divided by 2, for example. When you're doing this, try to think of where putting the parameter definition would be most sensible for somebody who's trying to reuse the model or try to read it. And it's always a good idea to annotate your specification blocks and also annotate the meaning of your parameters. And again, when we're coding rapidly in class, annotation may not be something that you want to do. But for example, if you're doing a class project, you would be expected to annotate all of your code and all of your parameters. Okay, so let's start actually trying to build avascular tumor simulation. And another thing about essentially good programming practice, which I strongly encourage people to follow both in class and also, and it's the way these exercises are designed in class, which is that they're sequential. You try to add one thing at a time to the exercise. I strongly encourage you to try to build simulations one step at a time, one method at a time, one component at a time. And so what we're going to do for the tumor is the first thing we're going to do is we're not going to let the cells grow, divide, or do any of the things that they really would do as cancer cells, because we're going to want to make sure that we understand the relationship between the cells and the chemical field. And that's why this is called a frozen tumor exercise. It's not going to be biologically very exciting, but it'll allow us to get a handle on what the parameter values need to be and how the cells and the fields interact. And so what we were going to do is we're going to create 256 by 256 lattice, use blob initializer to create a set of concentric layers of frozen cells. Make each cell 8 by 8. In the center of the lattice, create a disk of radius 40 that are necrotic cell type. Then a disk of radius 70 of quiescent cell type, and then a layer of proliferating cells of radius 100. And the trick is that in general, most of the XML, for example, the order in which you do declarations doesn't matter. But specifically, when you use blob initializer, the code is executed sequentially. And so if I actually draw the proliferating cell circle first, and then draw the quiescent cell circle, and then draw the necrotic cell circle in that order, I can make 3 concentric layers using blob initializer. I don't have to do anything fancy. In the diffusion field, we'll set the diffusion constant to be 30, which is pretty big, with no decay. Define a secretor plug-in for oxygen. Create a tracking field for the amount of oxygen taken up by each cell. And then in the Python steppable, define a secretor object that iterates over all cells and have them take up 0.1 of the oxygen with a maximum uptake rate of 1,000 and use the total amount version of uptake. And then run the simulation and see how the oxygen availability varies by position. Notice that you're going to have to, when you do the uptake, you're going to have to copy that into an amount taken up by cell dictionary entry so that you can display it. Okay. And so why don't people try doing this? You've done all of the things that we need to do here. Twit will do a lot of it, not all of it for you, but a lot of it. Alex, I don't know, you've been on the spot the whole time. Do you want to walk through this one as well, or
Sure.
how do you do it? Okay.
Part of it, but not all of it. Just trying to figure out the field, the tracking field.
Okay, so the first thing to do is, okay, so for the tracking field, you need to go into the init.
Oh.
And create the tracking field. So go to CC3D Python, extra fields, automatic tracking, track scalar attribute. There you go. And now you need to give the field a name. Okay. And that's about it. Let's see. Got to rearrange my screen a little bit so I can see you and the code at the same time. Okay, so you'll need to give that attribute name, something like oxygen again. Okay, that's fine. Now you don't need plot win. You don't need the plot win, although there's nothing. Well, you can leave that. That doesn't do any harm. And now when you do for cell, you do the uptake, which is fine. No, okay, so no, you don't. So line 33, you don't want. Get rid of 33. Okay. And now you don't need line 31 either. And so now you're going to do uptake. And when you do your uptake, you want the uptake, you're going to use the total count option. So if you go to the secretor options, I think you should have it there. Secretion, uptake, you want uptake inside cell total count. So you have to find the right one. Secretion uptake. So you have uptake inside cell. That'll be fine. And that one doesn't have total count. So you're going to have to add the word total count, which is fine. That's the one you have there we can use. So secretor uptake inside cell at boundary. And now you have to add the words total count. TOTAL. Yeah, there you go. And now we have to set something equal to that because we have to save it. So you could say result equals that. Okay. And now those other two we don't need. Okay. And now it's unfortunately total amount is the result. Result.total underscore amount will be the number you want. Result dot.
Maybe capitalized total.
Let me. That one I have to look up myself. Let's see. Let me show you the code here. So the code you need is, and again, it's in the manuals. So I do recommend people looking at the online manuals. They do help sometimes. Would be nice if they were actually also available in the right click in the help function. But result equals self.secretor uptake inside cell total count is what you need there. And then the amount that you care about will be, actually, I think I'm missing something that you're going to need here. it's going to be, let's see, it's going to be result dot total underscore amount is the answer that you, is the number you want. And you'll have to attach that now to a cell dictionary for the tracking field. So you'll have to have cell dot dict and then the name of the, what you called it, for example, oxygen. Equals result dot total amount. And there's a minus sign that you need. Yeah. Try that out. Alex, do you want to screen share and we can work on that together then? Again, if you don't remember the name, use that dir function on the object. So do you want to, okay, so that should work. Try that, see what happens. No, total amount is wrong. My mistake. So let's try during result and see what the correct name is. So why don't you do a print before that line, before line 37, do print parenthesis dir parenthesis result. Okay, there you go. And you're not going to want to do that too many times, so you can just hit step now. And we only hit step one more time, and then let's look at the output from that. You'll have to look in your Python console. There we go. And you see, dir will tell you all of the things that you have. It's TOT underscore amount, not total. The last one. So instead of total amount, it's TOT underscore amount. In general, I'm a big believer in not memorizing things if I don't have to. Okay, good. Now that's actually doing something, right? You're actually seeing the amount of chemical you're taking up per cell. And the amount taken up is varying between 0 and 26 in this case. Now let's look at the cell types. Can we look at the cell types? I only have, okay, you only have medium and proliferating cells at the moment. You haven't created the other cell layers yet. The other thing that you could see here is that if I'm understanding this right, essentially all of the chemical is being taken up in the surface. There isn't any in the middle. Is that right?
Yeah, I think I just have it set as only uptake at boundary. Right.
Okay, so we did something that we should be careful about, which is you created a tracking field, and the tracking field has got the same name as the chemical field of the diffuser, right? What's the diffusion field called?
I think it's oxygen with a capital O.
Okay, so I would call the tracking field something else like oxygen 2 to make sure that you don't, where you say field name also, I would give it a different name. It should be okay, but I would be a little nervous. There you go. And then change it also down in the field secretor is fine because that's oxygen. You change to oxygen too. There you go. Now try running. Because otherwise I don't know which one I'm displaying. This is actually the chemical field. Good. And you see the chemical isn't getting very far in. It's being basically absorbed all at the surface of the cells. All right. So now let's make the uptake amount less. So let's go into the simulation. And that uptake of 0.02 is a lot because that's per voxel. So you're taking up a lot of chemical there. So why don't we try making that 0.01 or something like that? And let's see what happens. Let's see how far the chemical gets into the tissue. It's still not getting in very far. It's basically only getting one cell diameter through. Okay. And now let's switch from the oxygen field to that oxygen 2 field to see how much actually the cells are taking up. So no, in player, leave it running in player. Just under the graphics window, switch to the other oxygen field. There we go. You see the surface cells are getting most of it, and then there's a little bit available inside. Okay. So that's not so bad. All right, now what we could do is we can create the other two cell types. And to do that, we're going to go into the blob initializer. And you've got proliferating, quiescent, and necrotic already. And so I'm wondering why those weren't showing in your cell type list. So let's come back to player. And go back to the cell field. And I'm not sure why. There we are. Okay. So I'm not sure why the cell type colors are not showing the other cells. That's, but they're there. Oh, there they are. Okay. So for some reason the cell type color there, that's okay. Great. Okay. Great. So now let's run that. And let's look at the amount of oxygen taken up per cell. And why don't we create a second image? Click the button just under the window. Click just there. Yeah, that'll give you a new. Okay, and now you can make that into oxygen field. Make that the oxygen field. So you have to pull down under cell field. You have to pull it.
It goes on the other screen.
Oh yeah, that's because of the zoom. Unfortunately, it puts that pull down in a weird place. Okay. Great. And so now let's watch this for a little while. And we can get a sense of what's going on. The chemical field is basically equilibrated. Actually, we've got, let's see, the chemical concentration. what are our boundary conditions for the chemical field? We want probably constant value boundary conditions, which looks reasonable. Constant value, that looks fine. So now let's go back to player. Yeah, max is 10, min is 0. That's fine. And the chemical is going about 1 cell diameter into the tissue. So we're still going to need to reduce the uptake a little bit more because we want the chemical to get a little bit further. What was the diffusion constant you were using? Okay, so the global diffusion constant is 1. Okay, so you've done something here that was, you've got the diffusion constant in your cells is 0.1 instead of, so you need to get rid of all of that specialization there. Yes, there you go. Now try it. Sometimes all that flexibility makes life a little more confusing. All right, now let's see what we got. There we go. So now maybe our uptake is actually too big. Yeah, so our uptake rate is now too small. In other words, now the oxygen's getting all the way to the center. And so now we have to make our uptake a little bigger. So try 0.1. There you go. And we'll let this run for a few minutes to equilibrate. And now what we can do is have the cell type change. We could say that the blue region, if the cells are blue, that is, they're taking up between 0 and 4, we want them to become necrotic. If they're taking up around 7, we want them to become quiescent. And if they're taking up 10 or more, we want them to become proliferating. Okay? And that's basically going to be our last exercise here. And I'll put that on the screen for a second for people. So now what I want people to do is the following. You've measured how much oxygen each cell is getting from the simulation, from what you just saw. So you want to make a note of that. And what you want now is to say that if the level of oxygen is less than half of the amount that's typically available for proliferating cells, the cell changes its type to become quiescent. If the level of oxygen is more than 2/3 times that, the cell goes back from quiescent to proliferating. If the oxygen level goes below, here I said 2/3, but you might make it lower, make the cell switch from quiescent to necrotic. Okay? Does that make sense now, Alex, given what you've got?
Let's try.
And then in principle, you can plot the number of cells of each type versus time and see how that changes. Now, in reality, the quiescent cells don't take up as much oxygen as the proliferating cells do. So we could change the uptake rates of the cells to be somewhat different, and then we'd have to change the thresholds to match. And I realized because of the issues we had with the people on the max. We've had to rush a little bit here, but I hope that the basic principle here is clear. We're going to create a fixed pattern of cells. We'll see how much oxygen is available under different circumstances. Based on that, we'll set a set of thresholds that allow us to change the cell types. And then we'll know how that behaves. And then once we've done that, we can have the cells actually divide and die so that we can separate the cell type changes and the diffusion behavior from the growth and death behavior. Okay. So Alex, I don't know if you want to show your code again, your simulation running again, since you've been so generous with that so far. Okay. So let's take a look at this. This simulation, let's just look at the amount of oxygen that's available and make some, you'll probably have to write this on a piece of paper. So if we want the outer two layers of the cells to be proliferating, we'll say that cells go from quiescent to proliferating if the available oxygen is above 10. Okay. And then we'll say the cells go from being quiescent to necrotic if the available oxygen, let's see where our blue is below 4. Just reading from the picture. And then the last thing we have to do is say that if the available oxygen is above 10, the cells go from quiescent to proliferating. Okay? And so then let's take a look at your Python and see about putting those changes in. So let's say if it's less than 4, we're going to go, the cell type will become necrotic. So you would say cell.type equals self.necrotic. And that one has to be all caps. That's the one that has to be all caps. Okay. And in this case, we better check because it only applies to quiescent cells. So we say if result total amount is less than 4 and
You type out use the ampersand.
I always use just and, but I think it's double ampersand in Python. Self.quiescent. Okay, that would be fine. And then we want to do the same thing. If the cell is proliferating and the level is lower than, we said 8, I think, we want to make it quiescent. Then we make it quiescent. And the last one is if it's quiescent and the level is above 10, we make it proliferating. And the one thing we probably want to do is we probably want to wait a few hundred Monte Carlo steps before we do this, because otherwise we have to wait for the oxygen to get into the simulation. So we might check to see if MCS is greater than 400 or something like that. All right, so let's see whether that works. What happens? And so now if we watch the cell type, yeah, the cell field should now become interesting. So let's actually look at the cell field one now. And so now we actually have emergent, quiescent, necrotic, and proliferating cells. At least once we get past 400 monoclonals. If we've tuned it right. Maybe we didn't have to wait 400, maybe 200 would have been enough, but let's just let it run. You said 400 was your time step to start? Alex? Oops, attribute error.
So proliferate.
Typo.
That's
Oops. And let's set MCS to 200 because I think it's equilibrated after 2. And it's wrong on line 47 too.
Still the other ones right.
Check your XML and make sure. Yes, and proliferating out, that should be okay. All right. And 20, 30, 40. You think it's going to work? It's going to crash? What do you think?
Hoping it works.
You're very close to a reasonably decent cancer simulation with this, actually. Once you've got these levels tuned, making it into actually a dynamic simulation like the one I showed at the beginning of class is not hard. Yep, everybody became necrotic. So what happened? Why did all of our cells become necrotic? We started out with almost everybody proliferating. Oh, I know why. No, let's see. If result, that total mass less than 4 and cell type distance. This should be okay. Huh. Okay. Well, I think I'm going to have to leave it at that. We're out of time for today. Anybody have a suggestion for how to fix it? For that one?
Is it with the result? Does it need to be negative result? Like
Oh, did we not put the minus sign in? I thought we did.
I think that was in the dictionary. It's there, but it's not in the...
Oh, well, that okay. Sorry about that. There you go.
Maybe that will work.
Yeah, there. Thank you. Yes, they're all negative. Thank you. That's good. Good catch. I usually actually, when I do that simulation myself, I do it off the dictionary entry. That way I know it's consistent. But let's see. But this is one reason that you want to build these things step by step, because if you try to build the entire avascular tuber spheroid simulation in one step, and you make a mistake like that, tracking it down, yeah, see now, look, you see the thickness of the layers has changed, huh? And it's pretty stable. So now I could change the levels of those triggers and change the thickness of my layers. So you've got it working. Great. Okay. So I think that's a good place to stop. That code is available in the student materials folder under the frozen tumor simulation. The next thing that we would do, and I'll give you a little bit of a homework assignment on that, would be to actually have the cells divide if they're proliferating, grow and divide if they're proliferating, and shrink and disappear if they're necrotic. So I appreciate people's patience. For people who have problems with their Macs, please screenshot the problem and document it and send that in, and we'll pass that on to Maciek. The code for the demos that we went through today is available online, and we'll try to make appointments to meet with people to talk about issues and projects later in the week. Okay. Any questions or comments before we break for tonight? I appreciate people's patience with the glitches, and I appreciate Alex. I really appreciate you being the one who was demoing everything today. That was very kind of you.